Good morning.
Okay, let's start. What we are looking at today is basically starting a new unit, which is after the completion of the previous history of medicine. What we are looking at is basically starting the unit, which is infectious diseases. So what you see on your screen is the unit specification, and this particular unit has 5 learning outcomes. We'll be going into a bit of unit introduction today, followed by discussion on the learning outcome 1, via presentation. We'll cover some of the assessment criteria as well. This is one of the main units that you're going to be studying in the nursing and midwifery course. And this is, a graded unit. So it is important that most of you are able to score, a higher or highest grade distinction, because it's seen when you do apply for your nursing qualification. Some units in particular over the others are given precedence by the university, and what they would want to see is that you have not just passed this unit, but you've achieved a merit or a distinction. So I will show you the assignment brief as well, just very briefly, to put a bit of focus in terms of how we are going to cover the assignment a bit later, but not going into the assignment in detail, but just looking at showing you briefly what the assignment would involve and what the assignment essentially would cover. So just briefly, for a second or so, showing the assignment brief. So this is going to be the assignment brief, which is available on Moodle. This particular unit is a core unit in biology, because when you do study nursing, biology and healthcare studies are two specific subject disciplines in which some of the units are divided. The next unit that you are going to look at is the aspects of human physiology, and that will also be covered under biology. And these are key subjects for nursing. That is the reason why you're doing this particular course as against a health and social care course. Now, this assignment is going to be 3,500 words cumulative. There are 2 parts to the assignment. There is part A, which is primarily an assignment that you will do. There are some tasks in it. And there is part B, which is basically a controlled assessment assignment, which is where you're allowed to, obviously use books, notes, or essentially bring your notes in or books in. But this will be done primarily as a controlled assessment. That means you'll be looking at sitting in a classroom with us here in Manchester and obviously writing the assignment. You would be able to access your notes and books, but the assignment is going to be primarily a handwritten assignment. So we will discuss the metrics of this towards the end of the learning discussions, learning outcome discussions, when we've completed learning outcome 5, and then we'll fix a particular day, for the week. And what we're going to do in that particular day for a couple of weeks, we'll be looking at calling it to the center. And the program essentially in that would be to take you through some of the practical aspects of this particular unit. So we have 10 different instruments, which mostly all nurses use when you work as a nurse within a hospital or within a medical setup. And they typically include an octoscope, which is normally for looking into the ear. You have a stethoscope, which is primarily to look at, your breathing, and, your heartbeat from chest, oblique from the back. We have a blood glucose monitor. We have an oximeter, which is to check your oxygen levels in the blood. We have a microscope, an electron, microscope, which is primarily, and along with that, we have 120 different culture slides that we will be asking you to go through. And these slides would be essentially slides that we have bought, which basically show different forms of, organisms, bacteria, viruses, and cultures. And it will be good for you to go through some of these under a microscope and go through these to understand, what are cultures and how, obviously, when research happens in this field of medicine, how it leads to, say, for example, looking at infections, looking at viruses, bacteriums, fungi under the microscope, and how do we isolate the genomes to be able to create medication for that. But here the idea would be just like in the biology lab in A levels or in GCSEs, we've understood and we've seen a lot of cultures under the microscope. That is what you're expected to do with us, at the center. So along with that, we will have a couple of other charts that we have, which are basically for all the human body systems, things like a skeleton system, digestive system, circulatory system, cardiovascular system. So the idea would be to try and go through some of these by looking at them and having a group discussion to discuss some of these aspects which are going to be related to some of the units that you're studying in nursing. So this will require you to come into the center 3 or 4 weeks on a particular day for about 2 to 3 hours, and that would mean that you'll be able to gain some practical experience on the usage of these devices by obviously using them and trialing them out, but at the same time also looking at some of these instruments and using them to gain a bit of a hands-on experience of what nurses actually use when you do work within the hospital. So things like taking bloods, looking at temperature, thermometer, looking at taking a patient's temperature, basic things that nurses would do to reach a diagnosis, or obviously look at bloods and other types of tests that we do, and they would be all instruments that we have that we will give you access to. Now, during one of those days, we are also going to be looking at doing this controlled assessment exam, and that would be, so the paper is already available, and that would be looking at this part B. So part A is basically a standard set of questions that we do like we do in any other unit. You have questions or tasks which are set related to the assessment criteria, and you answer those tasks. Part B is going to be a 2-part exam, which will be basically looking at understanding what is immunity, different types of immunity, and then looking at this diagram and interpreting it in terms of, how basically draw up a table and interpreting it to explain how the humoral immunity actually kicks in when you take medication, or when you are under, say, for example, an influence of a virus, or when you are falling sick. So the assignment is going to be interesting one, because this is the only unit in which we have this assignment, or the unit which has the assignment broken down into 2 parts, and one of them is like a controlled examination, and the other part is primarily an assignment that you do just as a creation of evidence, as a Word document. So we will be looking at discussing this in a bit more detail in a later session, which will be the assignment discussion session. For now, we are going to be concentrating primarily on, looking at going into the unit and understanding infectious diseases. Now, if I just take you back to the unit specification, what we are also looking at here is that when we look at the learning outcomes, we are primarily not just about covering some part of the content, which is given here. So things like bacteria, viruses, fungi, protozoa, and macroparasites, these are the ones that we are going to provide cultures for when you are able to see them under the microscope. Obviously, some of the viruses or some of the diseases which are highly transferable, they won't be available. So these are not something which we'll be able to provide, because obviously, there is no way any college, any university would have access to these cultures, because they can, lead to transmission. But most of the others that we get to see, you'd be able to see some part of the culture of different types of these microorganisms within, when you look at those slides with us in the center. Now, the unit is quite detailed. There is some part of it which will require self-study, and the reason I say this right now is because obviously on the Moodle you have access to a couple of handouts and obviously bits that I've created because this is the 4th or 5th time we are delivering this particular course. But the reason I mention this right now is that for those of us who have not had maybe not studied biology, during our GCSEs and A levels, or have not had biology as a subject, then in this case, there might be a bit of a reading that I would prescribe that you would need to do, is to bring yourself up to date to understand some of the basic concepts of things that we are going to discuss in this particular unit. So things like different types of organisms, single cell, multi-cell organisms, when we talk about how these organisms cause infection or diseases and what is their contribution and where do they sit in the ecosystem. When we talk about, different types of animals and animal systems, we talk about, we are obviously humans or mammals, but below us are birds, then we have amphibia, we have reptiles. And if we go down where the origins of life actually started, it all started with single-celled organisms, and that is where some part of this would need to be, you would need to bring yourself up to date in terms of your knowledge, and that is where this process of reading will come in. So on the Moodle right now, if I just quickly switch you over and show you this, I would make this unit active right now. And this unit is something which will have the yellow colored box, and this should be something which all of you should be able to access. Now on the Moodle, it's just very briefly switching over. So as we go along discussing lectures and obviously discussing the various learning outcomes, as you can see here, we've got the unit specification which I've opened up, which is what you get to see on your screen, which is given here so that you are also able to access it. Then after that is what we have is primarily a workbook. And this workbook essentially is provided by Ascentis. It's a 30-page workbook. Some part of the presentations that you would see primarily will have content coming in from this workbook. If you don't get to read any other book related to this particular module, this is one that you would need to kind of, go through in detail, because this will be very, very specific to the content covered in the unit. Although it might not give you, let's say, broader knowledge, but it will be focused on providing you the knowledge which is actually meant to be studied, and you need to know at least the bare minimums, and that would be given in this particular workbook. So this is an Ascentis workbook, very concise, very filtered, very good in terms of summarized content that you would be able to go through. And this is going to help you refresh some of the concepts that we have studied, in our biology days when you've studied that in schools. Things like types of blood cells, right? Red blood cells, white blood cells, platelets, type of how is the cell composed of, which is the powerhouse of the cell, mitochondria. We look at where the genetic codes of this is actually stored. So you look at the DNA, the ribonucleic acid, RNA, and those are the basic concepts that you will be able to go through when you look at this particular workbook. Now there are one or two other books or extracts that I've got which primarily will be useful for you to study, because they are a part of molecular biology, and because we are looking at studying diseases, we need to understand, and I will go into a bit of detail to understand terminology, what is infection, what is diseases, but how they are caused and what is the reason for the diseases or infections which humans pick up is going to be, in these summarized handouts that I've created or essentially downloaded, to basically help you understand the indicative content of the unit, and also to provide some additional information with regards to what is required to be studied, and for at the end of it, the knowledge that you need to gain, by looking at obviously the study of this particular unit. So these are extracted chapters from the book, which will essentially give you a bit more detailed information than the slides. Some part of this information from these handouts has also been used to create the PowerPoint slides, and that is where they would be useful for you to start reading now as we start studying the unit. Then we've got the 5 presentations that you've already, that are already uploaded here. And these are presentations that you can go through upfront so that we can have a bit more interaction during the sessions that we do, if you plan to do so like that. And then towards the end, we have the assignment brief, which is what I've just given you a glimpse of, just to, signpost it right now, not going into details, but we'll do the assignment discussion session at a later stage. Now, so that is how we are looking at essentially going into the coverage of this particular unit. What I'm going to do is obviously start our discussions today with regards to a bit of introduction to some of the key terminology that we are going to cover, with regards to this particular course and obviously this particular unit. And then we'll go into some of the assessment criteria as we start looking at these slides in a bit more detail. Any questions that you have at any given point in time, you can obviously ask me through the chat route, raise your hand. You can stop me there and then. And the idea would be to make sure that whatever I'm teaching or delivering, you are able to understand. And if there are any bits which are required for clarification, feel free to ask. Now, we'll go into some of the basic details of things like, the two key words in the unit, what is infection and what is, disease. So I normally look at a bit of a key terminology because these are bits that we are going to, use time and again. So when I talk about what is infection and I need to talk about what is disease, these are two things that we need to understand before we get into, this particular unit. Now, when we talk about, key definitions, let me put it this way. So if I talk about, say, for example, what is infection, we have lots of different ways through which we can define the word infection, but infection tends to be a complex biological process which involves microorganisms. Now, when we talk about microorganisms, what do we mean by that? So some of them say, what are microorganisms? One simple definition of microorganisms are, I would put it this way, is things that we cannot see with our naked eye, as simple as that, for which you need to either look at a microscope, use a microscope to be able to look at them. And they would be, bacteria, virus, they would be anything which is micro. Micro means small. They would be fungi and they would be parasites. So these are the 4 or 5 different types that we will generally see that are termed as what are called microorganisms. Now, why are they microorganisms? They are single-celled. In most cases, that means one cell which has the nucleus, the the powerhouse of the cell, which is mitochondria, and they would essentially be carrying out all the functions of an organism. So in our case, when we look at the human body, we are a multicellular organism. Our body consists of 14, 15 different types of systems. So when we eat food, there's digestive system. When the blood flows in our body, there is a circulatory system. When we breathe, there's cardiovascular system. The body also has an excretory system, respiratory system, lots of different systems that we have. We have a central nervous system, we have a peripheral nervous system. Now, these are systems which basically control the actions of a body, and that is why we are a very developed form of organism. That is why we are multicellular organisms, and we are basically at the top of the animal world when we look at mammals being at the top of the food chain. Now, when we talk about the microorganisms, they are the very basic building blocks of where we saw the origins of life actually happening. So single-celled organisms essentially, developed, and they are basically responsible for spreading of infection. They do not, they will essentially live off a host. So in this case, some of these organisms that we talk about, when we talk about, they living off the host, then in those cases, what we do is we talk about, organisms which are called microscopic or microorganisms. Microscopic or microbes or microorganisms cannot be seen by naked eye. We need to see them under a microscope. And the study of microorganisms is called microbiology. That means somebody who specializes in understanding how microorganisms live, breed, and infect other species. Then somebody who's going to be dealing with those studies, or as a criteria in terms of understanding the details of it, would be a microbiologist or somebody who's studying these kind of single-celled organisms in detail. Now, microorganisms are 4 or 5 different types. So you could have bacteria, fungi, you could have protozoa, which is another word that we normally, or another type that we normally have, which is again single-celled. And then there are parasites. So parasites are going to be classified as microorganisms, but they can be seen by the naked eye. So mosquito is a parasite that you can see. It sits on your skin and obviously draws blood. You look at leech. When you go to, for example, tropical climates or rainforest, you would normally see leech, as a microorganism which sticks to your skin, and they basically suck blood. You look at tapeworms, worms or hookworms, ringworms, which primarily human body can get infected with. they are organisms that you're able to see. So they are termed as parasites. They live on the host body, and they would draw food, energy, or any sort of nutrients that is required for them to grow, develop, expand, replicate, reproduce, will actually come from the host body. So when we talk about microorganisms, what we are looking at essentially is organisms which are also referred to as microbes. These are microorganisms which cannot be seen by the naked eye. You would need a microscope to view them, and they are tiny enough, but they are having the basic essentials of being termed as having life. So they are so small, but they yet cannot let's say we cannot see them in the human eye, but they would still be good enough for us to get infections or essentially catch diseases. Now, they transmit, or they kind of can what I'm trying to say is they can go from one body to the other, one organism to the other, or one host to the other, and in some cases they would essentially cause very, very let's say serious diseases in humans in particular. So when we talk about parasites, when we talk about microorganisms, say for example I start sneezing, and it could be a reaction that I'm having to a microorganism which has entered my body through the nasal route, which is my nose. And sometimes during summers, when we have high content of pollen in the air, what sometimes people do get to do is they start having what is called hay fever. And for hay fever, you normally get peritones. That means sometimes people can have a running nose, they can sneeze, they can get some fever, and for that you would normally be prescribed peritones to be able to overcome that. So you're allergic. In this case, your body is susceptible to these let's say microorganisms which can cause some sort of a reaction in your body, and that reaction would be an allergic reaction. Another example of that could be if you have fever, and that fever is not going away for 2-3 days, then you normally end up going to your GP, and the GP would ask you to get blood tests done. And when they analyze the bloods or basically the cultures, they then isolate is something is some sort of a microorganism infected the body. And in those cases, sometimes they would look at, yes, they would find that, okay, there is an infection, you have some respiratory infection, or you have a chest infection, and that has led, for your body to react. And obviously, that is why you're showing symptoms of having fever or coughing or sneezing. And in those cases, then the GP would, after the blood test reports have come, they would then recommend you appropriate medication. And that would be to help you get rid of that infection. So when we talk about how people get infections, they would be looking at understanding, the concept of how microorganisms would be infecting human body. Now, one of the other things that we want to look at, so one thing we know very clearly now is that when you get an infection, the infection leads to a disease or a symptom that your body will show. It could be, say, for example, you get a mosquito bite, and then you start scratching your skin, you get a bit of a blister or a bit of a swelling happening where the mosquito has, from where the mosquito is, you've had a mosquito bite on your skin. Now, that is your skin's reaction to primarily an external pathogen or a host which is trying to, inject something into your body, and your body cells, your white blood cells, your platelets essentially, start, let's say, fighting that particular external, agent, or in this case, the pathogen. And that would be a microorganism which has got, say, for example, transmitted into, your bloodstream, and your body is trying to fight that. Now, microorganisms cause infection. Infections occur in the human body when viruses, bacteria, or any other type of microbe enter your body and they begin to multiply. Now, this particular process would be termed as what is called infection, because a microorganism from outside has got injected into your body, and that has started a reaction, and that would be termed as infection. Now, what is a disease? A disease is something typically which will happen at a point in time when this infection begins to grow. That means your body is unable to fight the infection or the injected microorganism, and then your body needs external help. But before this external help in the form of medication comes to you, you would start to show symptoms of what is called a disease. And this would be a fever, high fever. Body temperature is normally 37.5 degrees. But if you get infected by a microorganism, then in those cases, what will happen is your body, you would start having high fever. You would start shivering, for example. And in those cases, what it is showing is symptoms of that you are getting a disease. So a disease occurs when cells in your body are damaged as a result of this infection, and the signs and symptoms of illness start to appear. This would be the process of what we would call infection. So when I say infection, occurs when viruses, bacteria, and other microbes enter your body and they begin to multiply. What would be a disease? A disease would typically happen in a small proportion of infected people, because sometimes our bodies are resilient and we are able to develop immunity. You might not show any reaction, but in some cases, when a small proportion of people with lesser form of immunity or weaker form of immunity start to show reactions, wherein the cells in your body are becoming damaged, and as a result of that infection or the microorganism which has entered your body, you start showing signs and symptoms of illness, then that would be termed as what is called disease. So these are basic things that we need to be looking at when we go into the unit infectious diseases. Now, there can be very many different types of infections depending on which particular microorganism is actually infecting your body. If it is, sometimes we hear the word, it's a viral infection. Sometimes we hear the word, it's a fungal infection. Sometimes we say it's a bacterial infection. Now, or when we also say it's a parasitic infestation or infection. So what we are basically doing is, when depending on the type of microorganism which is infecting you or affecting your body, the infection will also be classified on that particular type. And they could be viral, bacterial, fungal, or parasitic infections. Now, sometimes we say, when we look at, we talk about, say, for example, in the news we hear, the agricultural crops have been infected by moths, or there is a large-scale infestation which has happened in the plantation of tomatoes. Now, that is because a number of parasites or a swarm of parasites have actually infected the whole thing. And similarly, for a human body, when we talk about, the infections, if it happens at a scale wherein not one human or a lot of people are affected, then in those cases, you would either call them an endemic, pandemic. So we've gone through a COVID pandemic right now, which means it is something which has affected all humans globally, all the human population, across the globe. And that is why it was a pandemic. But when it started off, it was primarily an endemic, because the endemic essentially meant that the disease was occurring, but within a particular community. So it started in the Wuhan province in China, but as people traveled for the Chinese New Year from Wuhan to Italy and other places, the infection started to spread. And then this was not restricted to a certain area or a certain community. And slowly and gradually, what we saw was that this infection became global. And that is why the word endemic changed to pandemic from a WHO perception. WHO definition point of view. So when we had these regular briefings by the medical director of United Nations, they changed the classification of the endemic to pandemic, because people or populations across the globe started to get infected with the COVID-19 virus. And that is where you saw that large scale swathes of population were essentially affected. And that led to what is being termed, or what was termed as, a pandemic. That means it is present and it is affecting all communities across the globe, and it's spreading geographically. So widespread occurrences of an infection or infectious diseases, which could cover a whole country or a geography, or slowly and gradually the world, then in those cases, that particular concept is called the pandemic. A pandemic can be an endemic of an infectious diseases, but if it is spread across to a smaller region within one community, then we term it as endemic. But as it begins to spread and spreads across multiple continents, multiple countries, then in those cases, the definition changes to a pandemic. Now, with this basic understanding of looking at infection, how infection occurs, what are the various types of infection. Now, next thing that we want to also put into context is what so let me put it this way, how does infection spread? Any ideas? I've used the word here, but how do you think the infection spreads? If, for example, we talk about the COVID-19 virus, because obviously it was a virus which essentially, caused the pandemic, but how does the infection spread? So, yeah, go on.
It can spread through hands.
Yes. So transfer absolutely is the right word. The word is transmission. So transmission of microorganisms, which could be done through, when sometimes, I don't know if you recall, during our biology days, I've studied, like you have airborne diseases, you have waterborne diseases, and you have, when we talk about, how diseases or how infections can spread, we talk about, airborne. We talk about they can be transmitted through air, they can be transmitted through water, and they can also be transmitted through soil. So there are, different types of ways through which diseases or infections can actually spread. So sometimes when we look at, talking about how infection spreads, we talk about these kind of three different routes through which we see the transmission of the microorganism happening from one person to the other. And that process through which this particular thing, in terms of transmission of disease or infection, happens is basically called the process of transmission. That means infections can spread through various means. These means could be airborne transmission. When I sneeze, I could, we don't see anything in the naked eye, but when we look at sneezing, there are when you sneeze, it could be leading to you wheezing out, say sputum, which is your saliva, infection through if you have a running nose, into the air, which you normally are not able to see, but it can infect other humans because it can cause some sort of reaction in them, and the microorganisms then can, go into or enter into that particular body. So if this happens through air, then it will be called airborne transmission. If it happens through touch, then it will be called direct transmission. Sometimes when we talk about diseases, and I think all of us have had kids, so kids, and when I was growing up, everybody gets chicken pox, as one of the diseases. And one of the things that we do in terms, when your kids or somebody has chicken pox, is we do isolation. We put that person in one particular room, and there's limited contact, because the transmission of that disease can happen through direct contact. So in some cases, when we looked at the COVID-19 virus, the reason of having social distancing, face masks, use of face masks and gloves, and the basic equipment that we talk about, the gloves, the aprons, the face mask, and other things that we talk about, were essentially done, PPE as we call it, in the medical terms, was prescribed because what we wanted to do was minimize the transmission of infection. So personal protective equipment that we talk about essentially was used to minimize the transmission of COVID-19 virus, and that was done primarily to reduce transmission, which was happening from one human to the other. Now, excuse me. So when we talk about transmission or spreading of infection could also happen by ingestion. That means you can get infection sometimes when blood transfusion happens. And I don't know how many of you recall, in the news a couple of months back, there has been a very long-standing case which has been running against the government, is that in the 1970s and 80s, when blood transfusion was done, a lot of people got contaminated blood, and that was not properly tested, and that led to a lot of diseases and obviously serious conditions, patients losing their life. So relatives of those patients had filed a case, and I think after about 31 or 33 or 34 years, the case was won, and the government has decided to now give compensation. So transmission of diseases can happen, or infection can happen, through ingestion as well. Now, blood transfusion is one of the ways through which if you drink or have injected contaminated blood, then that can also lead to diseases. So diseases like HIV virus, we talk about some of the sexually transmitted diseases, they are basically transmitted through direct contact. But some of these can also be transmitted through what is called ingestion through contamination. And if blood transfusion happens from a person which has some sort of a condition already to a healthy person, then they can also get infection through the transmission of blood transfer or blood transfusion. So there are various modes of transmission of infections and diseases, and they can be dependent on the type of microorganisms which are responsible for that infection. Last but not the least, how do doctors, medics, practitioners essentially look at reducing the spread of infection? And that is normally done by prevention. So one of the things that we see for the prevention of spread of COVID-19 virus was essentially done through the use of what is called personal protective equipment. So all care homes, all medics, all practitioners within hospitals, any sort of a medical setup which were dealing with patients having COVID-19 virus, they were asked to basically wash their hands for 20 seconds, make sure they were wearing PPE or using personal protective equipment. And that was essentially looking at basically preventing, or to a certain extent helping in prevention of the spread of the virus. Now there are other ways through which we look at prevention. One of the other ways is, all of us have had the Pfizer Moderna vaccine. So vaccination is another route through which you prevent what is called spread of infection or diseases. Now a lot of us during our childhood days normally get that 3 drops of polio, rubella, mumps, and smallpox, and there are doses that are given to you a baby when the baby is 4 weeks old, 8 weeks old, and 12 weeks old. So you get 2 doses of that, and then what they are injecting by giving you those doses are the weakened microorganism or the inactive or passive microorganisms which helps your body develop immunity. So you never get smallpox. Smallpox is now eradicated out from the earth. You don't have polio and other kind of conditions happening in developed economies, although in developing countries it is still a major problem. And that is why the humans children at a very, very young age, at the age of 6 months, or I think 12 weeks or 6 months, are given this vaccination primarily to help the body develop immunity, and that is basically to prevent any sort of infections and diseases developing at a later stage. Now prevention is important because it helps in controlling the transmission or the transmission routes through which infections actually spread, and that is why prevention is at the foremost importance when we look at medicine in particular. It is a concept which basically looks at preventing the spread of diseases by using tools and techniques that are established right now to be able to say, for example, limit the spread of infections. So in general, when we look at the COVID-19 virus, hygiene, practicing good hygiene was essentially mentioned. That means washing off your hands, making sure if you've been outside and if you've had contact with open surfaces like in malls, high street, or escalators or places which are busy, in those cases it was recommended that you maintain your hygiene by using a sanitizer or washing your hands to ensure the prevention of a spread of this infection. So understanding transmission routes and employing preventive measures are vital in controlling the spread of infection. That is why prevention is also an important process when we talk about infectious diseases. So is this bit slightly a trip down your school memory lane to your school, but is this bit clear enough?
Yes.
Okay, now let's look at one of the things which is infectious diseases. So when we talk about, now we know what is infection, we know what is a disease. Now let's bring the term together and talk about what is infectious diseases. So infectious diseases are basically disorders caused by microorganisms. They could be bacteria, viruses, fungi, or parasite, and they live in our bodies, in or on our bodies. So when we look at mosquito, it doesn't cause an infection on when it bites on our skin. It can lead to an infection on our skin because your body might show a reaction and you might have a blister. But effectively, what it does is the Anopheles mosquito. There are lots of mosquitoes, but there's a very specific mosquito which basically causes, malarial fever. So when we talk about the Anopheles mosquito, that mosquito essentially is the mosquito which has the larva. It carries that larva which basically causes malarial fever, and that is fatal. And that fever, if you're not treated within the usual 10 to 14 days of the gestation period of the bite, and the Anopheles mosquito injects that bacteria, the microorganism into your body, and if you're not treated and the medication is not given, then it can also lead to death. So Anopheles mosquito is the mosquito which basically causes malarial fever. There are 460 species of mosquitoes, and most of them are over 100 of them are harmless. But there are certain species which carry this particular, infectious, larva, which takes 10 to 14 days to develop when injected into a body. And that injection happens through because the mosquito actually carries the live culture, and then when it sucks blood from the human body, it basically in return injects that larva into the human bloodstream. So there are going to be microorganisms which live on your body, which is basically your skin, and there are microorganisms which can be inside the human body as well, which can cause infection. So when we get worms in our body, tapeworms, ringworm, hookworms, and you have worms in your body, that means sometimes you feel that you have a stomach ache, or for example you feel bloated. And what generally tends to happen is that when you're asked to test for bloods, the bloodstream is able to pick up some of these microorganisms, and then that helps the doctor or the medics to diagnose that you have worms in your body, and that is where they give you a worm medicine to basically get rid of the conditions. In some cases it is not just once, you have to undertake a course for 3 days, 5 days, or 7 days to be able to get rid of the infestation, depending on how late or how early that has been detected. Now, infectious diseases can spread through various ways, and that is why the word transmission was something which I mentioned earlier. Now, this can be through direct transfer, it can be through air, it can be through water, it can be through ingesting into your body, or it can be also through direct contact with any other human or any other organism wherein the organism is carrying that disease. Now, when we look at bird flu virus, which is called the SARS-2 bird flu virus, as we call it, we look at COVID-19. These were viruses which were predominantly, the human body was immune to these viruses, and they were predominantly carried by different organisms. Like the COVID-19 virus, as we understand, came from bats, and consumption of bats in the Chinese community, especially in Wuhan, led to contamination, and that is where the virus actually jumped from being just in the bats to humans. When we look at bird flu, it is a virus which normally chickens and certain types of bird species actually carry, but contact with them, an unhygienic way of let's say maintaining a contact with some of them, can actually help these viruses jump to the human body. So these are conditions through which what we see is the transfer happening of the virus or the infection essentially from one organism to the other. So when we do get this through direct transfer, it can be through touch, kiss, cough, sneeze, and then is when the person gets actually infected. Now, there are various examples of how infectious diseases can actually transfer. So hepatitis A, hepatitis B, hepatitis C, novel coronavirus, or as we call it, the COVID-19, influenza, which is basically a flu. You look at measles, meningococcal disease. These are all diseases which are basically spread. They can spread from direct transfer of humans from one human to the other. And because they can spread so quickly, and they can cause the entire swathes of population to get infected over a small period of time. That is why they are called infectious diseases. And in such cases, any such medical program or any such medical facility essentially will forewarn people about that, okay, if this has been detected, then others need to, exercise caution, and you need to go into a mode wherein you would look at prevention so that you're not able to essentially, get that disease. And that is why when we talk about infectious diseases, they tend to be, spreading quite quickly through high swathes of population, and that is why some of these diseases can then develop into what are called endemics or pandemics. Now, flu, for example, a lot of people over the age of 60s in the UK during winters get flu because their body immunity goes down and they are susceptible to getting flu. And that is why the flu jab is normally recommended for people over the age of 50, over the age of 60, if they are also known to have what are called underlying, conditions. For example, they are diabetic or they have some sort of disability which would bring their body immunity down. And in those cases, these people would become more vulnerable, and transmission of diseases can happen quite quickly from them to other family members. And that is where the medics, your surgery, your social community nurses, or so, people who generally provide support for elderly at your home, especially if they have underlying conditions like diabetes or heart conditions, or any sort of other condition, they would essentially be advised that, okay, you need to be now taking precautions because of the fact that this infection can spread to other members in the family as well. So infectious diseases are a category of, I would say, disorders which can be spread by various organisms. They generally tend to be bacteria, viruses, fungi, and parasites. These microorganisms can invade or enter the human body, and they start causing what is called illness. That means I start to show symptoms of being sick or being not well. And they can also be transmitted from one person to the other. And this transmission typically happens through either contaminated food, water, it could be by direct contact, or it could also involve, for example, the example of Anopheles mosquito that I've given is also termed in the biological term as called vectors. Now, vectors are carriers or agents which carry the virus without the microorganism affecting the carrier itself. So if you see mosquito carries this particular larva, but the mosquito itself is not affected by the or does not get the disease. So it is a carrier, and that carrier is called a vector, wherein you could be carrying, for example, a microorganism or some sort of an infection in your body, but your body is immune to it, and you act as a vector, which is basically a carrier. And when people get in touch with you or get in contact with you, they can get the infection because their body does not have immunity. So that term is sometimes also called as a vector. Vector or carriers essentially are people who are immune to those diseases, because the host, as a host, you are immune, but you are still carrying the infection or the live culture of that infection. But when you get in touch with somebody else can get affected, but your body does not get affected because your body has developed immunity. So, excuse me. So when we talk about infectious diseases, we need to keep in mind that they can be transmitted from one person to the other. It can go from air, water, which are transmission types, and also through vectors, which are mosquitoes, for example, is one such example. And they would then cause what is called diseases in humans. Now, there can be different types of infectious diseases. Now, these diseases, as I mentioned, can be bacterial, viral, parasitical, or fungi. One of the things that you look at when I say fungi is something, we eat mushrooms. Mushroom is a form of fungi. Now, there are, I think, if I'm not wrong, if my memory serves me right, there are maybe hundreds of different types of species of, fungus. So if I look at mushrooms that we eat a lot of different types of mushrooms, button mushroom, penny bun mushrooms, things like that, which you normally buy from the supermarket. But that is basically a fungi. The biological term for mushrooms is fungi. Some of them are edible, some of them are non-edible, some of them are poisonous. Now, when we look at a disease called athletic foot, I know some of us have children and our children play football, if they don't change their socks regularly, or if their feet remain and shoes remain moist and they are wet, it can lead to a disease or a fungal infection, as we call it, and that is called the athlete foot, which basically leads to the accumulation of what is called a skin infection, which is caused by fungi. Now, how does fungi grow? Fungi grows in moist and damp conditions. We see bread. Most of us buy bread from the market. And when we buy bread from the market, what we see is if you leave the bread outside for a couple of days, you start to see green molds appearing on your bread. They are basically what are called fungi. Now, eating those can actually lead to food poisoning. So in some cases, when we look at the infections which are caused or diseases which are caused by fungi, we are aware, but I don't know whether it sometimes doesn't strike us that okay, athlete foot is caused by a fungi, not by a bacterium or a virus. When we see the green molds on our bread, and if it's a few days old, it leads to the growth of fungi on the bread, and that would be if eaten can lead to food poisoning. So these are kind of infections which are caused by fungi, which basically are also a type of a parasite, and they can affect the humans if we say, for example, do not maintain proper hygiene. Now, when we talk about infectious diseases, I also want to introduce a word called contagious diseases. So what is contagious? Contagious, when I look at the meaning of the word contagious, what I mean by the word contagious is it's a communicable disease. That means it can spread rapidly from one person to the other by direct contact. And COVID-19 virus, sexually transmitted diseases, as we call them STD, are very good examples of what are called contagious diseases. That means they are communicable diseases which can spread from one person to the other. And this spread generally happens through direct contact. You are coming in direct contact with a person which has the disease, and physical contact with that person essentially can lead to then transmission. Obviously, indirect contact can also lead to transmission. Sometimes what happens, and this particular example of the case which I've given, the contamination which was caused by the blood transfusion in the early 80s was an indirect contact, because somewhere, somehow in the lab, when testing of the blood samples was being done, there was a mix-up which happened, and that led to a large-scale contamination of blood when it was being given to healthy patients, and the patients then got that disease due to indirect contact. They did not obviously know that the blood being given to them during operative procedures was, infected, but that was indirect contact. So sometimes you would also have contagious diseases spread through indirect contact, and these can be coughs, sneezes, it could be by inhaling or by injecting, and that would be something which is called contagious. That means communicable diseases. Is that okay? Now, when I talked about the communicable diseases, influenza or flu, when we talk about measles, when we talk about, say, for example, meningococcal infections, HIV, we talk about STD, these are diseases which are communicable diseases. They are basically diseases which can essentially be, communicated by person to person, and this would be generally done through direct physical contact. Is that okay? Is that clear so far?
Yeah.
Yeah.
Okay. Now let's get into some of the assessment criteria for learning outcome 1 after getting through this background. So the first one talks about define pathogen and classify different types of pathogen that can cause infectious diseases. So when we talk about pathogen in general, what do we mean by pathogen? Pathogen is a term which refers to a microorganism or an organism which essentially has the potential to cause a disease in a host. So when I talk about a pathogen, pathogen would be a general term that we would use for bacteria, fungi, virus, or protozoa, or any sort of a microcyte or a microorganism which can cause infection. Now, in some places, if I look at when you are asked to, say, for example, if you're not feeling well, and you go to the doctor and say, oh, I'm feeling a bit lazy, and I'm feeling a bit lethargic, I sometimes also get fever, headache. The first thing doctor will do is basically ask you a couple of questions And if he is able to diagnose or not diagnose the problem, then they would say, okay, we'll have to do some tests. And what are these tests? These are going to be blood tests. Now, the blood tests are done because it is going to trace and see traces of any sort of infection which has happened in your body. And that infection, which can be traced by doing the blood test, would essentially be looking at what is called pathogens. Because as of that stage, until the tests have been carried out and they've been conclusive, I cannot say that you have a bacterial infection or viral infection or a fungi infection or anything of sort of an infection. What we do is we are testing your bloods for basically pathogen. Now, when I think I'm pretty sure that some of you have used the term or heard the term called pathology lab. Have you heard of the term called pathology lab?
Yes.
So what is a pathology lab? A pathology lab is a laboratory essentially of medicine or clinical tests or research which is carried out. And a pathology lab essentially looks at testing and doing analysis of your blood, urine, a stool, a tissue culture, or any sort of a diseased tissue that has been diagnosed as that has caused the disease. So when we talk about cancer treatment, when we talk about detection of cancer, the blood test, a tissue culture is taken, and that is sent to the pathology lab where it is tested by technicians who are basically experts in detecting and obviously carrying out tests to understand cultures of blood, and they would carry out these tests which are called pathology lab or laboratory. Now, they would be looking at analysis of blood, your urine sample, your stool sample, any sort of a tissue culture, and they will examine any sort of a, let's say a body part or a culture itself to diagnose a disease. And this part which is where it comes, it relates to pathogen. Because I don't know what is causing the infection right now. So I'll group them under one category and I'll call them what is called pathogen. Now, when I know that the infection is caused by bacteria, then I will go into details. The pathology lab or the pathologist, essentially the person who works in that lab, would then basically go into isolating that kind of a pathogen, whether it's a bacteria, whether it's a virus, whether it's a fungi, it's a protozoa, it's a micro parasite, and they will do further tests to isolate that pathogen. And then when I know it's a bacteria, I will go into further detailing to find out what kind of bacteria is it. So sometimes the government and the local council, as you know, issues warnings in summers that you should not swim into the rivers because there is high amount of contamination from sewage. Sometimes there were complaints, in Wales and this summer that a lot of water companies are actually sending, human waste directly into the ocean without having to treat them. And there was a lot of, protest against it during this summer. Now, why is it that they mention that don't go into open water reservoirs or don't swim in open places wherein the risk of contamination is high? Because in those cases, the water has or waterborne diseases, which means when you get in touch with contaminated water or water which has not been treated or not safe for human skin, can lead to contamination. For those of us who love swimming, one of the things that we see in the swimming pool that we put is chlorine, correct? So when you go to leisure centers, you normally see that, when before they start the season, they normally treat the water with chlorine and, certain other types of chemicals like ammonium sulfate and things like that. And that is to kill microorganisms in water, which could be infectious to the human body, isn't it? Right? So when we talk about pathogen is classified as, what is a pathogen? A pathogen essentially would be, say, for example, any type of a microorganism which has the ability to cause a disease. And once we analyze these in terms of by taking a blood sample, at some stage, what we will do is we would be looking at analyzing whether they are bacteria, they are viruses, they are, any sort of single-celled or other forms of organism, and they would essentially be then causing what is called the disease. Now, certain types of microparasite, I don't know in terms of when I was a kid, I used to get head lice because I have long hair. So Lice and ticks ticks you see in dogs or your pets. Lice is something that you see in the human scalp or your hair in the head, and you can get lice or lice infection if your hair is not, obviously maintained properly. People who have long beards can also get that, and these are examples. And lice is something that you can see with live eye. So I don't know if sometimes, during my childhood, I know when I had the infection, mom used to be able to use a fine comb through the hair and pick out, see the lices being picked out in that combing process. But today you get a medicine, and obviously I'm talking 30 years back, but today you get a medicine, you put it in your hair and obviously leave it for 15 minutes, then wash it, and you see in the sink or in your shower a lot of lice if you have had a lice infection. So microparasites sometimes can be seen by the human eye. Micro cannot be. The word is macro. Macro means you can be seeing that with the human eye. So lice is one such example. You look at ticks on your pets, dogs specifically get it, and you can see that, sometimes in order to take them off, you have to use a particular medicine or a fluid, and then it falls off from the body after a couple of minutes after that has been applied. And those are forms of pathogens which can actually cause infection. Is that okay?
Yeah.
Now, let's look at the structure of a pathogen. We want to go into understanding a structure of a pathogen. How does it look like? So what I've done is, in one of the slides, I've looked at the structure of bacteria, a basic bacteria, and what does it consist of in terms of its structure. So when we look at bacteria and we look at the structure of bacteria, what we are essentially looking at here is, we are talking about one cell, which is essentially, carrying out, it's a rigid structure which has some outer covering, inner covering, and that structure essentially, has the capability to carry out all the functions of the bacteria. So it will have different types of cells. The outer membrane is called the plasma membrane. Then you have a cell wall in which the cell is actually, the whole system of the bacteria is enclosed. You have what is called the plasma fluid. You have fimbriae, which is primarily the fibers on the outer wall of the skin, which allow it to attach to a host. You'll have some genetic material, which is called the chromosomal DNA. And then you have other forms of cells like ribosomes, mesosomes, and you have mitochondria, which will be the powerhouse of the cell. And this is what essentially will be, an example of a bacterial cell culture. That means it'll have some cell wall, it'll have some plasma, which is some sort of a liquid which protects portions of the cell from the outer portions of the membrane or the body of the bacteria, and it is usually enclosed in a wall or some sort of a layer. And then in some cases, some bacteria have what are called fimbriae, and these fimbriae are basically like tentacles or arms, which allow it to attach to a host. So when we look at lice, when we look at ticks, how do they attach? Or when we look at leech, for example, I don't know how many of you have been to a rainforest, but if you travel through a rainforest, you would normally see leeches attaching to the skins of, your legs or your arms, because they would basically use suction, and then at suction, they have suction pumps and small fimbriae which allow them to attach to your skin. And if you leave it for a couple of minutes, what they will do is then start penetrating your skin and start sucking blood. So they are, examples of how we would see, they attaching to the body of a human. So I don't know how many of you have seen a leech, but let me bring this up as a picture. And this is an example of a leech, which typically are found in the rainforests or typical rainforests. And if you go out for a nature holiday or some sort of a holiday, you would generally see that if the area has this as a, if it's quite wet and it's quite rainy, you normally have these organisms growing there, and they can attach to the human body and they can be seen by the naked eye. And this is an example of a macro parasite, not a micro, a macro, because you can see it with the naked eye. So this is an example of a leech. Now, the reason why we want to understand the structure of a bacteria or the structure of pathogens, bacteria, virus, for example, protozoa or fungi, is because we want to understand what is their structure and essentially what causes, the infection in the human body. So if I look at, putting in a slide, for fungi structure. And I bring just one up right now, just for the purposes of understanding what the fungi is and what a typical fungal cell is like. This would be an example of a typical fungal cell. So it will have what is called, an outer membrane. It'll have a cell wall. It'll have a nucleus. The nucleus is essentially, basically the main, let's say the main genomic part of the microorganism. This is what has the genetic code and the chromosomes, or the code essentially to run the cell and the cell functions. So in our case, we have nucleus in all our cells, but our central nervous system or our brain essentially does the coordination, because we are a very multi-celled, we are a multicellular organism, but we are also a very complex organism, and that is why we are at the top of the food chain. So the functions that our human body carries out requires not just a simple nucleus or a genetic code, but it requires a complex set of operations which is carried out by the central peripheral nervous system, and that is what works and connects and transmits all the signals from our brain down to our spine and other parts and organs of our body. So when we talk about a nucleus is basically a genomic cell which is enclosed in some sort of a membrane, and that cell contains what are called chromosomes, or the genes or chromosomes which contain the genetic code. And a nuclear membrane would allow certain type of material to pass in and out of the nucleus. So they are normally termed as proteins or nucleic acids. Now, when I talk about the various parts of the fungi, these are various components which basically make up the fungi. So you'll have a vacuole. I don't know how many of you remember the definition of vacuole, but vacuole is basically a cell, membrane-filled body, which basically carries all the waste products, which the waste is produced by the cell, and this then brings it to the membrane and then excretes it out or pushes it out by using the process of what is called immersion. So basically, the diffusion and immersion will allow the things to come into the cell and go out of the cell. So vacuole is like the excretory system of a single cell organism. Then you have mitochondria. Mitochondria is basically the powerhouse of the cell. It is the cell which basically produces, what you call energy for the cell to be able to, drive functions in it. You have cytoplasm, which is basically a gel-like fluid structure, and this essentially allows the cell to expand and grow and replicate. So this particular portion is like the zygote of the human embryo, which starts multiplying, and when it starts multiplying, it starts to grow, and the infection starts to grow, and then at some stage, this infection causes a disease, and your body starts to show some symptoms. So this is a typical, structure of, say, a fungi, which you normally see when we get fungal infections, and that is something that you need to be aware of. A bacterial cell will have more or less the similar kind of, let's say, components. But again, this is a typical bacterial cell, which is what you get to see under the microscope when you look at different forms of bacteria. Now, viruses are also very important because they cause, all sorts of viral infections. So when we talk about viruses, essentially, and the structure of virus, what we are going to be looking at primarily is, some sort of a basic, diagram, and that basic diagram of a virus would essentially consist of very similar things that you would normally have, with regards to, say, a bacteria, or say, for example, the fungi that we have looked at. So this is an example of the HIV virus structure. But in general, if I look at, various types of viruses and what they are consisting of and what they essentially are made up of, you would normally see that virus also has a very similar structure as what we see in the case of a fungi or a bacteria. But they are the difference between, sometimes you might ask a question, what is the difference between a bacteria and, a virus? So when we talk about a difference between a bacteria and virus, Bacteria are going to be single-celled organisms which can survive on their own. They can survive inside or outside the body, human body, or any sort of a pathogen body. Viruses will be basically causing infections by entering and then quickly multiplying within a host, and they would be multiplying to grow, their structure within within a particular host, and they would then essentially be looking at, causing infections within human body. So viruses would be just the genetic code, but bacteria would be a full cell which is able to carry independently its own functions. So virus would come into a body and infect because it's got the genetic code. It infects, it takes over a cell, and then starts multiplying with that cell by providing the genetic code. And that could be the easiest way that I could explain what are the difference between, virus and bacteria. Bacteria can multiply on its own. Virus would need the host to be able to reproduce. So what it will do is, it's like an intelligent being which carries the genetic code, and it will infect that into the cell, and then by giving instructions, it will ask that cell to multiply. It won't be able to multiply on its own. So viruses would typically have what are called ribosomes and RNA, which is basically carrying the genetic material. But in the case of bacteria, you would see generally all parts of the components being there in a cell, which include things like vacuole, mitochondria, cytoplasm, cell wall, membrane, and also the nucleus, which will have the genetic code. And they would then be the differences between these two types of or pathogens or microorganisms. And when viruses cause infection, that is called viral infection. One of the questions that I would ask you is cold a bacterial infection or a viral infection?
Viral infection.
So, anybody else?
Viral infection.
Okay. So, if you see symptoms that persist for maybe over a week or, more than a week or so, then in those cases, you would generally see this infection to be a viral infection, because viral infections, what typically they do is your body will fight the infection, and it'll at some point in time get immunity over it, because what it will do is basically fight the infection. And if what it tries to then do by fighting the infection is primarily start controlling the spread of the infection happening within the body through multiplication of cells, and that would then allow the the body to become immune. But if this thing does not get cured in a couple of days and it goes beyond a week, 2-week, 3-week period, then this infection would be a bacterial infection, and for which sometimes you would be prescribed what are called antibiotics. And because your body is not able to overcome this infection or develop immunity against it, then that is where you would normally be given external medication to be able to overcome those symptoms and to help your body develop immunity. So what we need to be able to understand is the first part of
Give me a ball.
If you could mute your microphones, please.
Thank you.
So what tends to happen in this case is that what you would need to do is understand the various structures of the for the first learning outcome, first assessment criteria, understand the different types of pathogens and understand the structure of the pathogen and how does it cause a disease. So we started with bacteria, then we go to viruses, we go to fungi, and that is what I have created slides for, to be able to understand and take you through that these are the various types of pathogens which can cause different types of diseases or reactions in the human body. And when they basically infect the body, they would then be essentially causing some sort of reaction, illness, or some sort of, let's say, you would start to see some signs and symptoms of it, which will essentially mean that your body is coming under attack from the pathogen, depending on the type of illness it produces or reaction it produces. And after the tests have been carried out, we will classify them into bacterial, viral, fungi, or any sort of macro parasite infections. So this part is to be looking at understanding the basic structure of fungi, virus, bacteria, and protozoa, and that would essentially help you understand, the first assessment criteria. Now, before I go into the second one, what I want to do is also talk about what is protozoa. Now, when we talk about protozoa, they are also single-celled organisms. They are also pathogens. They are classified as one of the pathogens which can, cause diseases. Now, protozoa are definitely microscopic. They are unicellular organisms, but they are also what are called eukaryotes. So there is a term called eukaryotes and prokaryotes. So I don't know whether you heard about these terms, but there's a term called eukaryotes and prokaryotes. So if I just, let me see if I've, okay, let me just put in a slide here and look at. So when I talk about these two types, there are two types which are called, one is called eukaryotes and the other is called prokaryotes. Now, when we talk about prokaryotes are always unicellular organisms, and eukaryotes can be multicellular organisms. That is what is the first, difference between the two. So when we talk about microorganisms can be unicellular and microorganisms can be multicellular. So when we talk about protozoa can be two different types of organisms. One could be eukaryotes, multicellular, and the other could be prokaryotes, which are unicellular. Now, eukaryotes can essentially be 100 to 10,000 times larger than the prokaryote cells. So if I look at the size, if I talk about the size of these organisms, they can be 100 to 10,000 times bigger than prokaryotes. They are still not visible to the human eye. Okay, so that is something that you need to keep in mind. We are still talking about microorganisms. Now, the DNA, which is the deoxyribonucleic acid, that is the full form, in eukaryotes, DNA is stored within the nucleus. So if we go back to the image wherein we talked about the nucleus, what we essentially did was we talked about the nucleus essentially having what is called the genetic material, and that would be the DNA essentially. And when we talk about the prokaryotes, here the genetic material, DNA, is stored in the cytoplasm, which is the film which I mentioned helps in the replication or reproduction of the cells when they start causing infection. So this is a difference between the two types of cells that we get to see when we talk about protozoa in particular. So when we talk about protozoa, we look at prokaryotes and eukaryotes. Eukaryotes they have a well-defined nucleus which is enclosed within a nuclear membrane, and the DNA, which is the genetic material, is organized into chromosomes. But when we talk about the prokaryotes, what we are looking at is that the DNA is actually loosely present, and that is present in the endoplasmic wall or the cytoplasm of the cell, and it is able to still carry out complex functions which would be required for the cell to survive. And that is where we would be looking at differentiating, these two types. So if I just put it slightly in this way so that you are aware, and I'm just going to create a bit of a wording here. So if I look at how do we differentiate between different types of protozoa, that is eukaryotes and prokaryotes, the way to differentiate them would be to primarily look at talking in terms of, number one, nuclear structure. Number two, talking in terms of cell size and complexity. And the third bit that we will be looking at would be looking at, talking about from a point of view of organelles, which is how thick is the membrane, or what are the various other cells that are contained essentially within the organism or protozoa. And then when we look at organelle, when we talk about the word organelles, we mean here eukaryotes will be contained in what is called a membrane, and that membrane will have all the cells available: nucleus, mitochondria, vacuoles. It will have Golgi apparatus, it will have cytoplasm, and it would be able to carry out independently all the functions. But when we look at prokaryotes, they will lack the outer membrane, and they will have minimum set of modules, which are like nucleus and mitochondria and maybe vacuole, to be able to perform certain functions. So when we look at the infections these two different types of cells in protozoa actually cause, eukaryotes cause major infections, prokaryotes cause infections, but they can be easily controlled by use of medication and obviously timely detection and prevention. So eukaryotes are typically found in multicellular organisms, and prokaryotes are found primarily within single-celled organisms like bacteria. So this classification that you need to look at is important, because what we want to understand is that when we talk about protozoa and when we talk about the microscopic, let's say, structure of how these are organized, we would need to get into understanding the protozoa from a point of view of two different types of cell structures. One is prokaryotes and the other is eukaryotes. So prokaryotes in general, single celled, if I may write it this way, and eukaryotes, because as you can see, they are 100 to 10,000 times bigger, they would be multi-celled. So if we look at, to a certain extent, the classification of protozoa, it is based on the characteristics and life cycle of what kind of cells would be infecting the host. And they are categorized into various groups, which include things like, again, going back to the school, we are looking at things like amoeba, or I don't know whether you recall the term amoeba, flagellates, and we talk about ciliates and sporozoans. And these type of cells, which basically have complex structures, can be single-celled or multiple-celled. So when I talk about amoeba, it is single-celled. When I talk about, for example, ciliates, multi-cell. When I talk about flagellates, multi-cell or two-cell. And when we talk about, other types of cells like the giardia or ant amoeba, they would be termed as multicellular organisms. So when you look at, now I will relate this and give you an example. So when you look at jellyfish, sometimes when you are on the beach and you see an announcement that this water could be infested by jellyfish. Is jellyfish, what is jellyfish? Any ideas? Any ideas? What is jellyfish? Single-celled or multi-celled? Any idea in terms of any classification? So jellyfish, if you see, they float around in the ocean with the ocean currents. They do not have any brain. They do not have any blood. They do not have heart. They are just very simple single-celled organisms. They have 2 or 3 layers in the body, and the outer layer is the tentacles, which you see floating around. The middle layer tends to be elastic, and then they have a bit of a jelly, which basically is called the mesoglia, which basically expands and contracts, which allows them to float in the water. So when we talk about jellyfish are basically what are called invertebrates. They have no bones in them. They are basically a part of, let's say their bodies are transparent. And when we look at what is their body composed of, it's composed of tentacles and, just basically simple arms, and they float around in the water. Now, they can be fatal in some cases, because jellyfishes can, sting and obviously be fatal to humans in particular. Now, when we look at talking about, some sort of single-celled and multi-celled organisms, we would then go into what is called the classification of eukaryotes and prokaryotes. And when we talk about protozoa, what we are basically doing is they are one type of pathogen which are called single-celled organisms, but they are different from bacteria. And they are basically classified as prokaryotes or eukaryotes. Now, when we look at the human body, and in general, when we look at certain types of protozoa which infect us and cause diseases, so when we talk about one of the STD, which is sexually transmitted diseases, that disease is called urogenital, disease, which means you have some sort of a genital infection in your urinary tract. Sometimes we are seen, you see people itching their bums, and that is a form of an infection which is caused by a protozoa. Now, what is that infection? It is basically a urogenital infection because it is in your urinary tract or place where you have your anus. And here, this is caused by what is called trichomonas. Now, trichomonas is basically a protozoa. It is not a bacterium. It is not a virus. It is not a fungi. It is a type of a microorganism which is called as protozoa. We also look at protozoal infections in our intestine, small and large intestine. Sometimes when you have dysentery, you know what is dysentery? Any idea what is dysentery?
Yes.
Have you heard about diarrhea?
Yes.
Okay, so what is dysentery?
Dysentery is normally when you go to toilet often or you have a stomach ache.
That is correct. So when you look at diarrhea, diarrhea tends to be a condition wherein you are frequently passing loose or watery stools. And this is caused by this basically is a cause of, if this is happening again and again, what will happen is this will lead to your intestines swelling up with some inflammation because they're having to repeatedly, process food and clear food, which is going into from your stomach to the colon to the intestine and then out of the body through excretion. So when you see this process happening again and again, this process is caused by a protozoa, which is called sometimes it is caused by what is called an antamoeba. And this process wherein you get infected and you go through the process of having stools again and again, or you have let's say serious condition wherein you are going and having watery stools again, this is called dysentery. Now, this is not caused by a bacteria. This is not caused by a fungi. This is not caused by a virus. This is caused by a protozoa. And that protozoa is called Antamoeba histolytica. And this causes a disease in the human body called dysentery. So when you look at malaria, malaria is also caused by a protozoa, which is actually carried by the Anopheles mosquito. It is not a bacterium, it is not a virus, it is not a fungi, and that carries the protozoa's larvae, and it is called Plasmodium. So these are things that you have to keep in mind when we talk about different single-celled organisms or multi-celled organisms, and that is an example of protozoa. Now, I mentioned the word Giardia protozoan. This is an important type of protozoan which causes a lot of infections in humans, and primarily is responsible for causing diarrhea and dysentery in the human body. This is the structure of that particular organism, which is called protozoa, and the protozoa name is Giardia protozoa, which causes dysentery or gastrointestinal infection in the human body. And that's the structure of the protozoa. Is that okay? Now, let's look at one last bit in terms of microparasites. So what are macroparasites? Macroparasites are, basically organisms that we can see with the naked eye. They are organisms which can cause, again, infections in the human body. They can be of different types. They can be lices. For example, when you get lice infection in your head or in your hair, in the, facial hair or in your scalp, you can get scabies. You can have tapeworms in your body or any sort of worm infection, and you can have ticks or microparasites, which can also infect sometimes humans and also our pets. Now, when we look at macroparasites, they include parasites which could be, classified into 4 different types. These are examples that I've given, but they are normally classified into what are called nematodes, tapeworms, flukes, and arthropods. So when we look at lice, it will be an arthropod. When we look at a fleece, when we look at ticks, they are basically, ectoparasites, and they can cause a bite. Sometimes we get to see and hear that if you have an old mattress, you might have, what do you call, some sort of parasites which could cause, bed bites. Have you heard this term? Bed bugs or bed bites? You can get rashes when you sleep on old mattresses or linen which is soiled or which has not been cleaned. So you get bed bugs as well.
Yeah. There's a bug. It's called bug.
That bed bug, very good, yes. So those are all examples of macroparasite. Macroparasites are things that we can see with the naked eye. They are type of germs or, essentially germs which are responsible for transmitting infections amongst humans. And they would be typically also sometimes transmittable from other organisms. For example, these microparasites can be transmitted through, if you are out and about in the field and you step on, say, bird poo, or you step on some sort of an infected, poo, then when you get back home and you don't wash and you don't hygiene or exercise proper hygiene, then this can latch onto the human body and cause infections. So when we look at roundworms and hookworms, they are transmitted through normally, open places where pigs actually feed, and they would then be causing infestation, or, they would be transmitted from such places, such open places where pigs and, farm animals are there, and they can transmit to the human body. So these are macro parasites, not micro. Micro is something which is not visible to the human eye. Macro is which can be seen by the human eye, and they are transmitted, and they can cause infections or some sort of infestation in a human body. And at some stage, you might not detect them immediately. Like children, sometimes they eat, sit in the ground and they can start eating mud. But over a point in time, you would not see symptoms of that happening, that they have developed worms in their body or worms have, but it is something that you will see over a point in time. It will not happen immediately. So in some cases, it will take 10, 15, 20 days, a month's time for you to understand that why is my child crying, or why is he having stomach aches and severe stomach aches. And then after that, when the bloods are done, they find out that, okay, the children has, they've eaten mud, or the contaminated hands when they were playing outside has led to the infection, and that has led to the development of worms in the body. And that is where your worm medication is then provided. So those are termed as macroparasite. So the first assessment criteria is all about understanding the term pathogen, the different types of pathogen that we talk about, 5 different types: bacteria, fungi, virus, protozoa, and macroparasites. And they have the ability to cause infections in human body. Is that okay? Now let's go across to the second assessment criteria, which is explain how the structure of an organism enables it to function as a pathogen. That means how does one of the type of microorganism actually cause disease in human, or humans start to show sign of illness or some sort of reaction in the body. Now this pathogen, which basically enters the human body, can cause infection, but infection can take time to show signs of illness, and over a point in time, this illness can become a disease if it is not treated. Now, there are specific pathogens which cause specific infections. So cholera, for example, is one such, disease which is normally caused by bacterium in humans, is an acute form of diarrheal disease. So cholera is basically when you drink contaminated water, or essentially when you are having water which has been contaminated with sewage or any sort of non-treated water, then this would cause diarrheal infections. And the bacterium responsible for doing and causing cholera as an outbreak, cholera normally spreads quite quickly because the contamination is waterborne. So basically you're ingesting water which has not been treated or has some sort of infection in it. And this is caused by bacterium called Vibrio cholerae. So these are types of infections which can be caused in the human body by essentially pathogens which are coming from outside, whether it's airborne, waterborne, direct contact, or through ingestion and they would then start to show what are called signs of illnesses. So let's look at how would a bacterial surface essentially cause, infection. Now, if I look at talking about Bacillus anthracis, this is one type of bacteria which basically causes, disease in the humans. Now, this is also termed as what is called anthrax. So anthrax is basically a very serious infectious disease which is caused by gram-positive and rod-shaped bacteria, and that bacteria is essentially called the Bacillus anthracis. Now, this bacteria can typically be, let's put it this way. How does it come in? Say, for example, it obviously is lives on soil. So if you have, say, for example, if you've gone out, you played, and your children sometimes when they go out, they play football and they're playing in a wet and muddy field, in a soggy field, and what tends to happen is when they come back, they don't wash their hands. What will then happen is they might have the chance that this bacteria is present and it can lead to some sort of infection. So the Bacillus anthracis or anthracis bacteria basically is something which is known to have what is called a protective capsule, and it has toxins in it which basically can evade the human body or the immune system in the human body. So you might carry this bacteria for a certain period of time, but at some stage you might not show any symptoms. But when the capsule actually explodes, it releases toxins into the human body, and this then can cause serious infection, which is called a disease, called anthrax. Now, when we look at the bacterial surface, and here we need to go back to this particular slide of understanding the bacterial structure. So if you see, there is a membrane outside the bacterial structure, and within the membrane there will be something called a toxin or a capsule, which will be enclosed in a capsule, the toxin. And when the bacteria actually explodes, or if, say, for example, it excretes, some, let's say, matter outside using the vacuole, it would then release the toxins in your blood. And these toxins, when they are released into the blood, can actually start by causing some sort of reaction on your skin. It might then, start to show certain signs that you are getting infected. And over a point in time, as the toxins multiply and obviously they enter your bloodstream, they can then cause, this disease, which is called the anthrax. Now, why do humans get Bacillus anthracis? This particular. Now, when you are out and about in, in the gardens, in the open spaces, sometimes what would happen is you will ingest or, breathe in spores of, let's say, bacteria. And, or if you eat something which is not being cleansed, or, it is not being properly hygienated, then in those cases, by eating contaminated meat, for example, steaks and beef and things like that, you would have this bacteria still having living in the meat, and then that bacteria will get into your body. And when it gets in touch with your body and your skin, obviously it then starts to cause infection, and that would be the reason why somebody would start developing a disease called anthrax. Now, this is one such example, because anthrax normally spreads from, beef, because essentially what tends to happen is that this bacteria lives dormantly on the cows and the skins of the cows, and when the wheat is cured and obviously stored, if it is not properly hygienic, say, for example, cured or chlorinated, sometimes you would have seen in movies, documentaries, that chicken, when it is processed, they normally run the chicken after, being processed, they run it through chlorine. And the reason why they run it through chlorine is to make sure that any sort of bacterial infection or bacteria which is living off the skin of the chickens normally gets, removed. So if the meat is not properly cured, what will then happen is this bacteria would survive on the meat. And obviously, when it is not properly cooked or properly cleaned, it will remain dormant or non-active. And at some stage, when it gets the favorable conditions of the human body, when it enters the human body, it starts to get nutrients and favorable conditions. It will then start to grow by releasing spores into your blood. And this will cause toxins. And these toxins then start showing symptoms of you developing this particular illness, because the cells have now become activated. From being dormant, they become activated. And when these cells become active, the bacteria starts multiplying, they start spreading in your body, they start producing what are called toxins or poisons, and this would start to cause severe illness. And if this illness is not treated, then this can also lead to death in humans. So this is an example of how you would see the structure of an organism enters the human body as a pathogen, and then after a point in time remaining dormant, it then at some stage basically causes what is called anthrax, or as a disease in the human body. So if I put this across just as an image on a new slide, you would probably see this as an example in terms of how this would happen. So anthrax disease and its transmission to humans via the pathogen Bacillus anthracis. So this is how it would basically go from remaining dormant on cow's skin when it is processed, then becoming active, and then it would start to multiply in the human body at some stage if the meat is not properly cured, and this would then start to lead to infections and finally to the development of a disease called anthrax. Now, this is the bit that we want to understand. How does a pathogen and what is the structure of that pathogen? In this case, we've discussed bacteria, and how does it transmit a disease to a human body? So it starts off by permeating, which is basically entering the skin, or by breaking down the permeative barriers that allows the selective absorption of nutrients. So at some stage, it will get absorbed in your bloodstream, and from there, when it starts to multiply, it basically would start producing toxins or poisons, and that would show the signs of illness happening in the human body. And the human body will try and fight it, obviously through your blood cells, your white blood cells and your platelets, and they would be basically fighting the pathogen. But in some cases, if it is not able to fight and it is overrun by the reactions caused by the pathogen, in this case the Bacillus anthracis, then what will happen is you would need to be treated, you would need to be given medicine, and if medicine is not given on time, it can also be fatal, and person can lose their life. So in some cases, detection of these diseases and the spread and the detection of these diseases and the timing of it needs to be very important and critical, and that is why some of these, when they enter the blood, that is why blood tests are recommended. That is why the blood cultures are then obviously analyzed in detail to understand what bacterium or what kind of pathogen is causing this infection, and then accordingly doctors give the medicine to treat that particular illness. Is that okay? So this brings us to the end of today's discussion as far as learning outcome 1 is concerned. Any questions on this discussion today?
No.
Okay.
Uh,
I have a question.
Yeah, go on.
Yes. I didn't see this on the module, but I've realized you've added it now. Is the assignment on it too?
So the Moodle has everything on it. Obviously, the presentation sometimes gets updated because I've added a few slides. So you will also be able to get the updated version of the presentation in a couple of minutes after the session.
Okay.
But Moodle has everything on it.
Oh, okay.
Yeah,
Thank you.
I want to ask about the books. Is it from the school got the book, or where?
So in your case, when you're looking at studying this particular unit, what I would suggest is, as I mentioned earlier, open this particular workbook, at least this particular workbook, which is provided by Ascentis, and it has some of the details that I've covered in the presentation today.
So I can open it on the Moodle.
Yes, that's correct. You can open it. For example, when I click on this, it is opened up. You can also download it and save it on your PC. So click on this download button and you can save it on your PC.
All right.
Okay. So
All right.
this presentation I've updated slightly, and what I will do is I will put this up on Moodle. It will replace the Learning Outcome 1 presentation in a couple of seconds. Okay.
Okay, thank you.
Thank you.
Good stuff. So we'll catch up tomorrow to discuss Learning Outcome 2. Until then, thanks for joining today. And then tomorrow we'll be discussing the second part of the learning outcome, which is LO2. And I will see you then.
All right, thank you.
Thank you very much.
Take
Bye.
care. Thank you. Bye.